Dear fourth-year student, my name is Fan Wang, and I'm from the Institute of Biomedical Material and Devices in the University of Technology, Sydney. So today I want to show you my team and my research. The reason why I want to show you this video is because I know many of you may confuse that why you need to learn such a lot of scientific knowledge during your degree, and also you may confuse that how you're going to use those knowledge. So today I'm going to show you what is scientific research looks like and what's the connection between those knowledge and the cutting edge research. Before showing my lab, I'm going to give you a little bit introduction of my research. My research focuses on the biophotonics. And photonics is a technology to manipulate photons, which is the smallest element of the light. And the biophotonics is using the photonics tool to study the biological problem. You may think we already have knife and a tweezer to do the biological experiment, why we still need to use the photonics tool. The reason is we want to see the small features. If you think the human body is a really complex working machine and we have lots of gears inside, and if you want to study what's the fundamental of life, you have to figure out what's the working function of the individual gear. You have to see the smallest gears. But the size of the gear is really small, like exosomes. So basically, it's got a size 1,000 times smaller than the cross-section of human hair. You have to use the photonics tool to see them. And in my research, I use my physics knowledge to generate a new technology to inspect those single gears and to figure out what's the working function. For example, you can track in how the single nanomedicine transferred inside a living cell. Traditional microscopy method enable us to see the small feature like bacteria, but if you really want to look at the single molecules inside of the bacteria, you have to use the super resolution technology, which is awarded by the 2014 Nobel Chemistry Prize. And my interest is working on the deep tissue super resolution technology. So we're interested in to see the single molecules through the deep biological tissue. This is the only way enable us to see the cell-to-cell interaction through the deep tissue in life. And this is my system and my PhD student on top. So we are working on this technology. If you look at the system, we got many of these optical gears, optical elements, and we can use them to build any optical system. In this technology, we use one special lens to generate one tube-like structure beam. So this is so-called second-order Bessel beam. And this beam have a really long penetration depth, can send this beam in through the deep tissue, reaching the point we want to resolve. And if you look at the cross-section of the beam, we got a really nice donut shape. And only the feature inside the donut can be resolved. And if you shrink the pinhole into a nanoscale set, then we have nanoscale resolution. For example, this is how we resolve nanoparticles with a size of 50 nanometer. This is my PhD student Paulette, and we are working on large field deep tissue super resolution imaging. So this technology provides you much faster imaging speed. And to reach in-depth imaging for large field, we use aberration correction technology. I give you an example. So for the first class, usually we couldn't see through that, and because the surface was getting lots of light. But if you put a sticky tape on top of that, you can compensate the aberration, and then you can see through the fluorescent glass. And here we can use a similar technology. We can use the beam shaper to compensate the aberration from the biological sample to reach faster in-depth super resolution imaging. Apart from just see the small feature, I'm also interested in using light to trap small particles, and this technology is called optical tweezer. And this technology is awarded by 2018 Nobel Physics Prize. So optical tweezer requires a strong focused laser light, and if you squeeze the light into a small volume like 1 micrometer, you can generate a really strong electromagnetic potential well. If your small particle close to this potential well, it will subject to the gradient force and come to the bottom of the potential well. And if you move your laser spot, and then you can move the particle. In my research, I'm focused on to generate the next generation force probe, which is a small nanoparticle but can provide 30 times enhancement on the optical force. So you can either use this particle like the first probe to measure the localized force change, or you can use it to dress a single cell to increase the interaction. So after the dress, you basically can make the cell run like the wind.
This is in my research. I hope this video will inspire you and make you interested in science. If you are interested in doing honor project or PhD with me, my email address is shown below.